You may have heard about how your cells have to be constantly dividing. But did you know that every time a cell in your body divides, it has to copy its entire genetic instruction without a single typo? Get it wrong, and you could end up with a mutation, a disease, or worse. So, how does it happen? Let's break down DNA replication step by step. Before we start to go into the process, let's just clarify a few things. What is DNA? DNA stands for deoxyribonucleic acid. Later on you will see that RNA stands for ribonucleic acid, so you just clip off that. DNA is a molecule that carries genetic instruction. I will later on show you how the structure of this allows for this. DNA replication is the process where cells copy its entire genome. This is basically how it creates a duplicate of itself, copying all the genetic information. This process occurs before cell division. This is important because during cell division, the cell makes a complete copy of itself. So it needs the DNA to have already been replicated to split into the new cells. DNA replication occurs in the nucleus in eukaryotes and in the cytoplasm in prokaryotes. This is because in prokaryotes, there is no defined nucleus, no membrane-bound nucleus, rather. Before we move on, remember that form equals function. So, before I start the process, I'll be explaining the structure of DNA, which will explain and will help remember and understand how this works. As you may know, DNA has a double helix structure. That is this shape over here. Each strand of this structure is made up of DNA nucleotides. A DNA nucleotide. DNA nucleotides are made out of nitrogenous bases, deoxyribose sugars, and a phosphate group, as you can see here. There are 4 different types of nitrogenous bases. You have thymine, guanine, cytosine, and adenine. And then later on when you learn about protein synthesis, you will see that thymine is often replaced with uracil. But that's for a later stage. It is important to know the base pairing rules. That is this here. Cytosine and thymine are under purines, and guanine and adenine are under pyrimidines. Cytosine and guanine bond together, and thymine and adenine bond together. Cytosine and guanine specifically bond with 3 hydrogen bonds. And remember, hydrogen bonds are weak. Thymine and adenine bond with 2 hydrogen bonds. Before I said that DNA is a molecule that carries the genetic information. Here's an example. As you can see, these are nitrogenous bases on one template strand of a DNA. In groups of 3, these are called codons. Codons are what relay the code. Instead of just taking every individual nucleotide base, it codes in groups of 3, called codons. Lastly, it's important to know that DNA replication is a semi-conservative model. What this means is that the DNA strand, the DNA strand will split into 2, meaning that if you took this red color as the original, it will split exactly into 2, so the 2 new DNA molecules that is formed will have exactly 1 strand of the original and exactly 1 strand of the new synthesized DNA. Okay, now after understanding the structure and what is DNA replication, we are going to go into the basic high school level outline of DNA replication. This visual model is what I will be using to explain DNA replication on this type of level. Okay, I like to split my processes into steps so it's easier to learn. So it'll go into unwinding, unzipping, using a template strand to make a complementary strand, and then forming a new model. So let's start from the beginning. As we know, this is how DNA base pairing works, right? I tried to make the colors match so it'll be easier to see, but don't mind that. So firstly, we know that this is how the original DNA molecule looks. It will unwind, which means the double helix structure will untwist, as we can see over here. This untwisting causes the weak hydrogen bonds to unzip, as we can see over here. Next, these strands here, which come from the original DNA strand, act as a template. Therefore, this template strand on these sides will help to form the new strand, which happens over here, which is complementary, which, as you can see, means that the nucleotide bases will match up. These nucleotides over here that come in are free nucleotides that are found in the nucleoplasm. Because remember, this is all happening in the nucleus, which means that these nucleotide bases will all come from the nucleoplasm. Lastly, the new molecule will form, which has exactly one strand of the original DNA and one new synthesized strand, therefore making this a semi-conservative model. So now that you understand the basic outline at a high school level, we are going to build onto this to form a more proper and accurate biological level explanation of DNA replication. Now, before you freak out, let me just give you an overview of how we jumped from this to this. So here we used this really nice visual model of a DNA molecule. We are just going to zoom in to this section over here, because this is where all the complexities happen. This is a more in-depth look at a DNA replication model. Over here, we have the DNA strands. This is one strand and this is one strand. The dark gray will be from the original DNA strand, while the light gray over here are the new nucleotides that come from the nucleoplasm. I've taken out, which should be over here, where the DNA molecule unwinds its double helix structure. But that I feel like is very basic to understand. But you can see here, this over here is the original DNA molecule with its two gray bits, which are the two original strands. It splits this way and this way, and the new nucleotides, this light gray, gets attached. So you can see here, there are two different DNA molecules. Okay, so let's dive in. Just to preface, we are going to be talking more about how all of these, as you can see in highlights, and all these little blobs, which are enzymes, how all these enzymes catalyze these steps that we were talking about. Before we build onto the original process, the basic process that we learned, let's just revisit the DNA structure. You will see in a bit that I refer to the different sides of a DNA strand as 5' and 3'. So here, one thing about DNA is that it has directionality. Okay? The nucleotides link via phosphodiester bonds, which creates the directionality. And we read this always as 5' to 3'. And these also run anti-parallel, as you can see here. This is important because oftentimes when I refer to processes, and when you will be in the future writing out these processes and describing it, you will describe that nucleotide bases join a template strand from, for example, the 3' end, and so forth. So, for example, this is a new synthesizing strand, right? Just pretend. And it is being created in this direction. We would say that the DNA nucleotides, which is this bit over here, are being added onto the new strand, this being the new strand, onto the 3' end. Because this is the 5' end over here, this is the 3' end of the new strand. So we would say that the new nucleotides are being added onto the 3' end, like that. But then again, someone could also say they are being added from the 3' end to the 5' end on the template strand. So this way, 3' end to 5' end on the original strand. That's why it is important for you to understand what the different primes mean and how the directionality is used when we are speaking about this process. Initiation. This occurs at the origin of replication, where the replication bubble and replication forks have formed. Firstly, helicase unwinds the DNA structure. So the double helix structure will be unwound because the helicase enzyme breaks the weak hydrogen bonds, as you can see here. Next, the single-stranded binding protein keeps this from rejoining, as you can see here by these two blobs. This one, helicase, breaks through it, almost like a zipper. And this single-stranded binding protein just jams in the way so these two can stop from joining together again. Next, topoisomerase enzyme relieves the tension from overwinding. So that is over here. This is important because as this replication bubble here is unwinding, it can cause the preceding DNA molecule to overwind. So the topoisomerase enzyme helps to break, swivel, and rejoin pieces to avoid strain. That is initiation done. Using these 3 enzymes, it helps to prep the DNA to start making the new molecule. Okay, elongation. This is basically the synthesis of the new strand. Firstly, it is important to note that DNA nucleotides can only attach to a nucleotide already attached to the template strand. For example, over here, a DNA nucleotide cannot just attach like this because it has nothing to latch onto. To combat this, short chains, about 5 to 10 nucleotides of RNA, which are called primers, this red things over here, which are synthesized by the enzyme primase. The DNA strand starts to form on the 3' end of the RNA primer. So that would be, this here is the 3' end, so this would be 5', and this here would be 3'. Therefore, where you set down the primer, which is synthesized by primase, the DNA nucleotides are attached to the primer, which help it attach to the original strand. Specifically in this example, I will be talking about DNA replication occurring in eukaryotes, which use DNA polymerase 3 and DNA polymerase 1. In prokaryotes, they use about 11 different DNA polymerases. So we've already spoken about primase and the primer. Next is DNA polymerase 3. This is what actually adds the nucleotides, the respective nucleotides. As we have mentioned before, the base pairing rules are very strict. So DNA polymerases use this template to add on the appropriate DNA nucleotides that respect the original. But the way that the DNA polymerases with the primer works is like this. Starting at the origin of replication, there is something called a leading strand, like this, which starts at the origin of replication using a primer and goes toward the replication fork. Okay? And then at the same time, but a little bit delayed, is the lagging strand, which work from the replication fork and toward the origin of replication, as we can see here. The lagging strand is also made up in segments, as you can see. These are called Okazawa fragments. So now you know how DNA polymerases work. Next, after the DNA polymerases and primers have all set it out, DNA polymerase 1 goes in and replaces each primer with the respective DNA nucleotide, because remember, the primer is just temporary. And then following that, once all the RNA primers are gone and everything has DNA nucleotides, the gaps that join these Okazari fragments, as well as join the leading strand to the lagging strand in the middle, are all joined together by DNA ligase, which is basically like glue. So, in all, initiation and elongation have prepped the DNA molecule to synthesize a new DNA molecule. You can also note for DNA polymerases 3, DNA polymerases catalyze the synthesis of a new DNA by adding nucleotides to the 3' end of a pre-existing chain. But how do they do this? Well, initially, nucleotide triphosphate, which look like this. A nucleotide attached to 3 phosphates. Lose 2 phosphate groups, and they are lost as a pyrophosphate. So these 2 phosphates are known as a pyrophosphate, which hydrolyzes to 2 molecules of inorganic phosphate. When you cleave off the pyrophosphate, this reaction is exergonic, which helps to combine the 2 nucleotides with the respective number of hydrogen bonds. Okay, now we've done initiation, we've done elongation. Before we go on to the last step, which is termination, let's just talk about repair. So, most cellular systems for repairing incorrect paired nucleotides due to the DNA damage or replication errors use a mechanism that takes advantage of base paired structure of DNA. So, DNA polymerases proofread the new DNA strand and replace the incorrect nucleotides using an exonuclease action. The reason that this whole step here is very important and why it comes before termination is because if it is not corrected before cell division, the mistake is permanent. There are 3 enzymes involved in this. So here in this example, I've said that the damaged section of DNA is due to UV. So a segment of the strand containing the damage is excised by a nuclease enzyme, as we can see here, which is nuclease is the DNA cutting enzyme. Next, this gap is filled by DNA polymerases, which fills it with the correct nucleotides. And then lastly, DNA ligase, which acts as the glue, repair all the gaps and seal all the free ends. Therefore, the DNA is successfully repaired, and it can move on to the termination step. Okay, now the last step of DNA replication, which is termination. This is basically the release of the 2 new DNA molecules, as well as closing any open ends. It is important to note that in eukaryotic chromosomes, they have linear DNA, whereas in prokaryotic chromosomes, they have circular DNA. The reason that the linear DNA in eukaryotes have a problem is because they cannot complete the 5' end, since there is no 3' end of a pre-existing polynucleotide to connect to. So here, these red are the RNA primers. When they are replaced by the respective DNA nucleotides, this bit here on the end, on the very last fragment, cannot be replaced by the respective DNA nucleotides because there is no pre-existing polynucleotide, like over here, for it to connect to. Therefore, in repeated rounds of replication, it produces shorter and shorter DNA molecules with staggered ends. So this bit here will just cleave off, which cuts it down to this, and then another round of replication will cleave this end off, and the molecule will just get shorter, which is a problem. And a problem that prokaryotes do not have because they are circular. So they will always be able to replace the RNA primers with the respective DNA nucleotides. So to fix this problem in eukaryotes, telomeres help. A telomere cap binds onto the last fragment to prevent the shortening that occurs. An important thing to note is that this specific sequence characterized by TTAGGG, which is repeated over 100 to 10,000 times, is non-coding. So this section in purple over here is non-coding. And this section over here is the actual coding DNA molecule. The reason this is so significant is because as DNA replication goes for multiple rounds, it may be cleaved off at any time over here, but it will not be affecting the genetic information because this section here is non-coding. So it protects the coding section of the DNA molecule. Okay, so now that you understand the process of DNA replication, why is this important and what is it used for? Well, the main thing DNA replication is essential for is cell division. During meiosis or mitosis, which is cell division, your cell also needs to copy and transfer genetic information for growth, tissue repair, and healing. So every time you've gotten a cut or bruise, your cells are rapidly dividing to try and heal the area. Transmission of genetic info. Did you know that you have exactly 50% of genetic information from your mother and 50% from your father? This process is really important to duplicate your parents' genetic information and hand it to you when you are born. High fidelity is essential for organisms to survive. High fidelity is basically the proofreading ability. And the proofreading ability of DNA helps to catch and fix any mistakes to avoid mutations which can cause disease and disorders. As I said before, DNA replication is a semi-conservative model which ensures stability. The new molecule will contain one original strand and one new, which reduces the chances of errors. So how is this process used? Well, this process is often used in cancer biology, chemotherapy, and cancer drug development. A big part of cancer, if you didn't know, is basically uncontrolled DNA replication, which can result in a tumor. It can also be used for antiviral and antibiotic drugs, as well as biotechnical applications, such as the COVID-19 test, forensic DNA fingerprints, and genetic disease screening. For example, over here, you may actually be given this in a test. You are given a DNA fingerprint test that is found at the crime scene, and you are given the suspect's DNA. You need to see what exactly lines up, so which suspect could be the one that was at the crime scene. This is very simplified and is not exactly used in the field, but oftentimes comes up in tests in high school. This here is gels. In labs, you can actually separate the different contents that are in a cell and extract the specific component that you need, such as DNA or maybe protein, and run tests on it. So this is an example of what that looks like. A big part of DNA replication also circles back to the cell cycle. As you can see in this very busy diagram, DNA replication plays an important part in keeping this cycle going, and it is an important part of what makes it functional. Okay, so now I'm going to go through a few practice questions that you can encounter going from a high school level to a tertiary education level. Okay, so I think the most obvious question you may be asked is, explain the process of DNA replication. Now, if you just learn the process and learn the structure that comes with it, this question should be the easiest that you encounter. So first of all, on a secondary school or high school level, these are the points I'd like you to touch. So what and where might get you both points, 1 mark and 1 mark. And then, like I said, you split the process into steps. In this case, I've split it into 5 steps. So unwind the double helix structure, making sure to drop that name. Might get you 1 mark. Then you unzipping specifically the hydrogen bonds which break. Might get you 1 mark. Then using this, the original DNA molecule as a template strand. Get you 1 mark. And then onto the template strand, you are using base pairing as well as the free nucleotides in the nucleoplasm to create a complementary strand. This might get you 2 marks. Lastly, you have a new molecule which is semi-conservative and consists of one original strand and one new strand. And again, the double helix structure retwists. This might get you 2 marks. So in total, that is about 9 marks, plus minus. Depending on your high school, it would be a good idea to check past exams and past tests to see what exactly they are looking for and what terms they are looking for. So for example, like double helix structure, hydrogen bonds, base pairing, nucleotides, semi-conservative, like markers like to see those specific words being used in the right context. Now, for tertiary education, you might have to touch on what and where, which will count as 1 mark together. And then getting into this process, it's good to break it down even more because it gets so complex. So in initiation, you can explain where this happens, the origin of replication, the replication bubble and fork, just to give the scenario where it's happening. And now whenever you drop the names of each enzyme, it is important to say what exactly they are and what they do. So for example, helicase breaks hydrogen bonds, single-stranded base proteins, single-stranded binding proteins keep the 2 strands open. Topoisomerase enzyme relieves overwinding. DNA polymerase 3 add in the nucleotides. The primase creates the RNA primer, and then you can explain what the RNA primer is and why you need it. Then you can explain how the leading and lagging strand forms, as well as dropping the name Okazawa fragment. Then you can explain how the DNA polymerase 1 replaces the RNA primers with the DNA nucleotide. And then lastly, DNA ligase comes in to join all the gaps. And then lastly, you can explain how termination happens and add in the fact that you need a telomere, which might get you 2 marks because that's a lengthy explanation. So in total, that is plus minus 13 marks. So in total, the main thing I would give you a tip for is to learn the processes by splitting it into these little templates and making sure that you hit all the specific terms they want you to use and use it in the right context. Okay. Oftentimes, I think more so in high school and secondary school, you'll be asked questions based on base pairing rules and Chargaff's rule. So, if you are given this DNA template strand, what is the complementary strand? So here we are seeing a 3' to 5' template strand that has this sequence. We know that the complementary strand will run anti-parallel, so from a 5' to a 3', right? And then the complementary code will lie in between this. So we also know that, well, for both of these rules, adenine and thymine bond together, and guanine and cytosine bond together, right? A and T, G and C. So here we know a T will bond with an A will bond with a T, a C will bond with a G, G's will bond with a C, C will bond with another G, A with T, and T with A. So that would be the newly synthesized complementary strands code. I think this part may be the easiest, but students will often forget to highlight the directionality. Because remember, the 2 strands will run anti-parallel. Okay, now for Chargaff's rule. This basically states that in DNA, the nitrogenous bases that are binded together will have equal amounts of each other. So the amount or percent of adenine will be equal to the amount of thymine, and the amount of guanine will be equal to the amount of cytosine. And obviously in a whole DNA molecule, it will just be 100% overall. So, cytosine, you have 30%, okay? Which immediately should make you think that, okay, cytosine bonds with guanine. And because of the Chargaff rule, these two have to be equal. Therefore, guanine also has to have 30%. Therefore, these two will equal together 60%, leaving 40%, because 100 minus 60 is 40, will equal to the amount of adenine plus the amount of thymine. Okay, but you also know that adenine and thymine have to be equal. Okay, this writing is very confusing now, but hopefully you're following along. So the 40% has to just divide. So there'll be 20% of adenine and 20% of thymine, because together it will equal to 40%. So you just divide by 2. Therefore, you answer the question, how much thymine? There is 20% thymine in this DNA strand. Easy, right? In tertiary education, the questions are oftentimes much more difficult and don't have a specific answer. So, there are 2 types of questions you could get. Recall and perspective questions. These type of questions force you to recall the processes and knowledge that you have learned, but force you to look at it from a different perspective. So they will not just flat out ask you, explain DNA replication for so many marks. Instead, they will say very specifically, compare prokaryotic DNA replication with eukaryotic DNA replication. Or maybe compare the role of DNA polymerase 3 versus DNA polymerase 1. So testing on DNA replication in different types of cells, or maybe the enzymes that are a part of DNA replication. Similarly, they could ask a purpose for a specific step in DNA replication, like what is the purpose of the origin of replication? Or they could ask you to explain what is the purpose of the end replication and the problem that eukaryotic chromosomes have during the termination phase. Things like that. However, the hardest questions that you usually get are the application questions, where instead of even asking you about DNA replication, they say, based on your knowledge of DNA replication, what happens if, and so on. They will use your knowledge of this process and see how you can apply it. So here's an example. A researcher treats bacteria cells with a drug that inhibits topoisomerase. Predict the effect on DNA replication. Now here it tests multiple things. Do you know that bacteria are prokaryotes? So therefore, if you do know that they are prokaryotes, do you know how DNA replication works specifically in prokaryotes? Next, they expect you to know topoisomerase's role in DNA replication. Therefore, knowing these two, what happens if topoisomerase was inhibited, which means that it is not being produced? So what is happening if this enzyme is basically removed? So if you remember, topoisomerase helps to relieve the overwinding of the DNA structure. So because a replication bubble opens and all these things are happening at the origin of replication, further along the DNA molecule, the strands could possibly twist too much and damage the DNA. So knowing this, if there was no topoisomerase to prevent this from happening, it is likely that the DNA strands can become damaged and can break. So this might have an effect on the genetic information that is passed on, and so on. This is why I say that in tertiary questions, there isn't exactly just one answer or one specific thing that they are looking for. They like to see how you can explain yourself and how you can use what you know to the best of your ability. So I think the best thing you can do is just to understand the process as much as you can and just come up with some answer that makes sense.